Thank you for attending. So I think we will have the questions after each speaker, and it's with great pleasure today that I introduce Professor Heike Grabsch. She's a UK-trained histopathologist specializing in gastrointestinal pathology, holding a chair at the University of Leeds, UK, and also at Maastricht University in the Netherlands. Professor Grabsch has a long-standing research focus in predictive and prognostic biomarkers for patients with gastric or esophageal cancer. She has published widely in this field together with international collaborators. I welcome Professor Grabsch to talk to us about predicting genetic alterations for pathology images with artificial intelligence.
Yeah, good morning, and thank you very much, Jimmy, for the invitation. It's great after 2 years of COVID to travel again, although I had forgotten how long it actually is to get here, how many hours you're sitting in a plane. I can see the slides there, but not here. Okay, thank you. So I'm talking this morning about predicting genetic alterations from pathology images with AI. And our big advantage during the COVID epidemic was that we had all these images. And so we didn't need to go to the lab. We weren't in that way closed down. And that has made quite some progress during that time. So if we're looking at AI, it's called the third revolution in pathology. We first had in the 1980s immunohistochemistry coming in. And of course, this was a big advantage. We could all of a sudden see proteins in the tissue sections. We then had in around 2010, next generation sequencing, which was called the second revolution. And about 2018, we are now able to analyze images in pathology. We are scanning images since 2000 or longer, but we were never able to actually analyze the whole slide images because of technical problems, because the computing power wasn't there. And so you can see also that, why is it not moving on? It's not moving. So you can see this in the publication, basically, how this is steeply gone up after 2018. And that has to do with the computing, that there were all of a sudden graphical processing units. And this is really a boom in publications. And this is only in the field of tumor pathology. So in other fields, they are not even recognized here. So what can we do with being able to analyze images on digitized slides? We can simply do fundamental methods. We can help finding things, quantifying immunohistochemistry. We can find the tumor. We can do tumor subtyping. What I will be focusing on today is to detect genetic alterations in H&E, so our standard histology slides. And this is not moving. Yeah, now, okay. So, but you may wonder, why would we want to do this? So why analyze images from histological slides if we can do molecular testing? And one of the assumptions, of course, is that any mutation, any copy number change leads to a specific change in the morphological phenotype. And if that is true, then of course we can see that there is a mutation, or the machine, the algorithm can see it. One major driver is the cost. Molecular testing is expensive, and not all of the laboratories can afford to do this regularly. But if you could see a molecular change on the image, on the normal routine H&E stained section, then everyone can do this. And so that is one of the main drivers. And the other driver is the speed. So if you have an H&E section, you can put it into a box and out comes the result. This patient has, I'm just saying something, a P53 mutation, and that's what you're interested in, then this is extremely quick, whereas otherwise you need to extract the DNA, go via sequencing, etc. And that could make the pre-selection or the screening of patients for definitive molecular testing quicker, but also the screening to, for example, send people with suspected hereditary cancer syndromes earlier to clinical genetics. So if you do this, so we published in 2020 in Nature Cancer using the TCGA data, as many people are using them, because the TCGA has the advantage, it has all the molecular data, but you can also download all the histology slides. And that is what many people have been doing now. And if you're using that data set, we focused on 14 major tumor types and used something that is called unsupervised deep learning. There are other methods that doesn't really matter. What you can see here, I think I can't point with this. You can see the results in this panel for lung, colorectal, breast, and gastric. And everything which is above this line, about 1, that is detectable. That means that leaves a signature, something I can see in the microscope. But it also means everything that is below that line, which is quite a lot, doesn't leave a footprint in the images. So I cannot see those. So that's the bad news, which I've listed at the bottom of the slide. Overall, we can only see or detect, predict 30%, 30% of all genetic changes make a change on the histology. The good news is that the 20 most common mutations, which we all are familiar with, including all the mutations which are targetable by FDA-approved drugs, known oncogenic drivers and signatures, they are all visible. So they have induced enough changes that an algorithm can detect them. And you can see this, for example, here, also that there is a difference by tumor type. So for colorectal cancer, we can link, in this case, the BRAF mutation to histology quite easily. And as if you are a pathologist, you can see quite how different the mutated BRAF cases are from the wild type. But in gastric cancer, which obviously is my favorite topic, unfortunately, gastric cancer is very difficult and very different. It doesn't work for gastric cancer as good as for colon cancer. And the relationship there with the highest driver mutations is not really so clear. So we cannot see all genetic changes, and although we can find, or the machine can find differences in the histology, for the human eye, I can't see that if I then compare the images. It's much more subtle. And yeah, I'm not sure. So right after, basically back to back, the other paper from a group was published, and they actually did the same thing. So we were basically had the same idea at the same time, and they basically found the same things. They extended this to not just mutations where we focused on, but they also looked for copy number changes, for amplifications, deletions, and gene expressions. And they also found that not all, only a subset of these genetic changes can be seen, and that it doesn't work in all cancers. And I think this is also important, that one shouldn't have the idea you can do everything, you can solve every problem with doing AI on histology slides. And what you can see here in the pie charts is that some of the changes can only be detected by AI on histology slides, others can only be detected on histology or on both. So it's quite variable here. So that was one too much. No, that's moving in the wrong direction. Yeah. Now it's not moving. Can we move on forward? One more, please. And one more. Yes, so the next aspect is, so well, if we cannot predict all of the molecular changes, does it give us any other advantage, apart from what I said already, cost and speed? Can we learn something from doing sort of molecular profile on the image? If you can move on, yes. So for that, I'm not sure how many are familiar how we actually do this. There has been obviously a big breakthrough on the computational side, but we are still not able to analyze the whole image, the whole H&E slides, which you can see on the left side, in one go. This is not possible because this is about, you have to think 2 or 4 gigabytes of data. And computational power is not big enough to do this in one go. So what we have to do is to cut the whole image into these small patches and analyze individual patches only. We cannot do the whole in one go. And the way it works is then that you will have an H&E from a case where you know it's, for example, p53 mutated, and you have obviously thousands or hundreds of those cases, and you have similarly a similar number of cases who are wild type. You're cutting them into patches. You recognize automatically where the tumor is located. That's the blue region that is outlined there. You put them in something which we call a convoluted neural network, and there are all sorts of different technologies that I think isn't important here. And what comes out here, what you can see on the right side is, for every patch, you're getting, here overlaid with color, the information is, does this phenotype in that patch seem to be related to a p53 mutation or to wild type? So everything which is red, the machine predicts in that position you have a p53 mutation, and everything that is blue, you have a p53 wild type. And if you seeing that, we can basically learn, have some additional information, and I'll show this in more detail with the TCGA data. We have all been, hopefully, have also seen the TCGA classification for gastric cancer, where we have the EBV subtype, the MSI subtype, genomically stable and chromosomal unstable, based on molecular features. So the question now is, these molecular 4 subtypes, can I see them in the image? So you're using the images and the molecular information. You are training the model. You need a second series to also test the model. And then the first challenge is, what, as I just showed to you, well, you're having now all these patches, and some of them, let's say, are positive for one type and negative for the others. What are you going to give to the label? What label are you going to give to the case? What people are doing at the moment, and that's for debate, is we just go for the majority. So as in the previous case, if the majority of the patches are red, then one would call this whole case has a p53 mutation. And so if I'll show you this, so at the top row, you see what we call the ground truth. That's the molecular data, so the 4 subtypes, SYN, EBV, MSI. But if you're looking at the sort of third row from above, you can see per case it actually varies. So you will have cases, in particular, if you focus on the GS, genomic stable. So there's only one case where the column is completely yellow. And all the other cases, they actually show also the other classifiers. And that means that actually there is molecular class heterogeneity. Of course, if you are extracting the DNA or the RNA, you will not know this because you have extracted the RNA of the whole case, not from individual regions. And the next slide shows this I'm just showing this in more detail. So here you have on the right-hand side a molecular defined chromosomal unstable case. But if you're looking at the individual patches, you can see there's also other phenotype in it. So we can actually all of a sudden see that the heterogeneity, we can localize the heterogeneity. And there's the same as another example for genomic stable, where you can see from the phenotype, you have all 4 types in there. This was then tested, so you're training the model on one series, and you validate on another. This is a series from Cologne, and you can see the same phenomenon. It's not uniform, the phenotype. It's heterogeneous, as we probably all suspected. What is interesting is that in the Cologne series, the MSI, so microsatellite instability status, could not be predicted on the H&E stain slide. And we similarly struggle with the prediction of MSI in gastric cancer. That's a real problem on the H&E. And we're still working on this, why this is the case. But here the take-home message is, using this type of methodology, we can all of a sudden really see the heterogeneity. We can see which molecular type we have at the surface, which molecular type we have in the depths of the tissue. And here is just to illustrate this. It's even clearer if you're only going for one change. So this is, again, the example of P53, where you can see here on the surface, on the right side, that all has the phenotype, predicted phenotype of being mutated. And at the, in the center of the tumor, it's all wild type. And that, of course, offers really further analysis. And that's also true for the consensus molecular subtype for colorectal cancer. So this is also TCGA data. So same procedure, you use the TCGA data to train the model to recognize a phenotype on the H&E, and you're testing, in this case, on the FOCUS trial and some material from Aberdeen. And it's the same story. You can again see, so you can again see the heterogeneity in the actual tissue, so that different molecular subtypes are present in the same tumor. We probably all knew this or suspected this, but now we can finally see this. And therefore the question is, or maybe it's an opportunity, So is this type of work, which we have done here with Rakav, Dr. Sanda, where we actually, separated the different regions and extracted by region. No, this was not to move on. Can you go back? Yeah. We did this spatial profiling. Do we still need to do this in the future, or we just do it on the H&E? And I think that is a possibility that we couldn't, in that study which we published in 2021, we could only afford to do about 70 cases because it's very expensive to do this. Can you please go back? And because of the costs, we were not able to do this on a large series. But now with this new technology, we are able to really investigate heterogeneity on a larger scale. In this study, we discovered that the deeper part of the tumor is more related to the lymph node metastasis. And those studies we can now do at a larger scale. The other opportunity we have here is that there are cases which we cannot classify molecularly, and because you don't have enough DNA or RNA, but if you do it on the image, all of a sudden you can classify them. In this case, it's the colon CMS typing. So you can study all of a sudden heterogeneity, and you can classify cases which you couldn't classify before. So does this now all have clinical utility? And one more. So just to show you the MSI work we did. So we were one of the first group who published in 2019 that you can predict microsatellite instability directly from the histology image. The first study was a relatively small study, and the result, which is measured as the area under the curve, was 0.84, not bad, but it was really one of the first papers. It was already clear at that time that it works much better in colon than in gastric. We then started to increase our numbers, and in the second study, we were already able to analyze more than 6,000 cases, and you can see already that the area under the curve increases. So the more cases you have for the training of the model, the better. And then in the and then in the last publication, we were able to move this up to 9 different colorectal cancer cohorts with more than 8,000 cases. We also tested this on biopsies and resections. And we were able to find out if we're setting the sensitivity to 95%, then up to 50% in each cohort were classified as microsatellite stable. And so our recommendation was then use the artificial intelligence to rule out microsatellite stable cases. If the artificial intelligence suggests MSI, so instability, then only do testing. And that saves you about 50% of the molecular testing. And our friends in the US did a calculation. Yeah, this is really not working well. And how much would that actually mean in cost and time? So if you're looking on the top right side, so if you would do, so these are US figures, so if you do next generation sequencing, it would cost you 3,500. Cost for PCR or immunohistochemistry was calculated about 1,200. So mismatch repair deficiency you do either by immunohistochemistry or by sequencing. But if you do it on the H&E, you only need to consider that you need to do the scanning. And the time is it's really in a few minutes. Where the time for the sequencing, they calculated with 12 days, and time for PCR with 4 days. And they basically built a model with 32,000 patients, and compared then different combinations of AI on H&E with immunohistochemistry, PCR, and next generation sequencing. And the essence of this is if you combine high sensitivity AI, so analyzing the H&E, the normal histology slides, which is there anyway, and only testing the ones where the machine predicts with high certainty this is a stable case, then you really can deliver accurate results with 97% diagnostic accuracy, very fast and cost effective. And we were pleased to see that it's a company, I'm not involved in that company, but since few days, or since the 2nd of September, this has been approved by the EMA. This is a list of the approved tools, and by either EMA or FDA, so you can use it now. It's now the question whether people will use it. And so if you as a clinician request, well, do it via this pathway or not. We have to see. There are pathologists I know will see, well, no, I don't believe it. I want to see the immunohistochemistry myself. So that will be the challenge. It's possible now. We have shown it. It's approved. But whether it's going to be used, we have to see. And then the last slide is just to wrap up. So yes, we can, with computational methods, predict known key molecular features from standard H&E sections, but by far not all of them. We can do it on biopsies and resection specimens. What also the image analysis gives us as pathologists an advantage, because it's really great if you can see all of a sudden that, yes, if a case with a P53 mutation has a different phenotype, I can see this. That is really an advantage for us, because you can pre-screen. And those things will come in more and being approved, I think, in the near future. It will still be, I predict, it will still be a challenge whether people are going to use it, because there is this black box where you're putting in your H&E slide, something is happening there, and then out comes this is microsatellite stable or not. And whether you believe it or not is the question, but also there has been the discussion who is responsible. So if the machine now says something and that's not true, and you are treating the patient based on that results, who is responsible for that? And that's a question that has not been resolved. Who signs that report? And so, no, if you as a clinician ask for it, you get the result directly. So we were going to push this to the clinical team, I think. So thank you very much.
Okay, thank you, Professor Heike. Do we have any questions from the audience first?
Is the code coming out, or?
Oh yes, the Slido code. There it is. There are no questions yet, but oh yes.
Thank you very much. I can see why this technology is going to be a threat to the livelihood of pathologists, and therefore be some reluctance. But my question relates to that comment you made about defining the tumor base that's got great heterogeneity based on the majority. I'd just like you to explore that with us a little bit, because clearly there may be an underrepresented portion of that, less than the majority, which is actually determining the ultimate fate. So what do you think we need to do here? Because you just make the assumption, the consensus definition of the majority. Where do we go with that? Because is it possible to define the prognostic significance of a minority population rather than just taking it from the majority visually?
Yeah, I think so. I think we now have the opportunity to investigate this further. If you're only doing the molecular test, you're only getting one number. It's positive or negative. Now we can see, well, not all of the tissue is, let's say, p53 mutated, but there is another portion which maybe is not mutated. And I think if you now have the right material available, you now can do these studies and can really see, okay, what is the influence of this heterogeneity? Is it that if whatever 20% of the tumor doesn't show this phenotype, that is really a difference? So we didn't have the opportunity before, or only if you would do spatial profiling. Because spatial profiling on the molecular side is extremely expensive. And
I just wondering what you can actually do. I was wondering, my mind went to whether in that one pixel or that one image, could you have a replication rate or something about the activity, the cell division rate, to be combined with?
Yeah, I mean, and that's the other part, obviously, that you can have now on the H&E sort of classifier, on the H&E section, you can overlay all these different classifiers. So you can do, for example, the proliferating part or the genome duplication part, and in the same sort of patch you would also see mutation or expression. You can overlay all these different ones and can analyze this now. So I think it's a great opportunity. Regarding the challenge to pathologists, I personally think it's great if a machine does all these boring things, measures for me, and ideally I would like to get rid of all the easy cases that a machine can do, and I can focus on research or doing more complicated
we still
things.
want you to sign the pathology report, though.
One question? Yeah, thanks. Yeah, Jimmy So from Singapore. I think Heike, this is really spectacular. Therefore, you are trying to use the artificial intelligence to extend the pathologist's capabilities.
Yeah.
Because we don't usually do the molecular testing using H&E stain. Yeah, because we also involved in artificial intelligence research. But most of us, we are trained the machine to learn what the human can do and try to duplicate or make it more efficient. And what you are trying to do is to extend the capability. And I just want to fall back a little bit. Can you just give us an update on the easier tasks in terms of AI in pathology, whether the AI can actually help the pathologist to read the H&E stain, the normal pathology report more efficiently? Because I think that not every hospital in the world have expert pathologists. In fact, we see a lot of overseas patients, the pathology report sometimes is wrong. Therefore, we get second opinion. Therefore, I thought this task is already quite an important task, whether AI can help to equilibrate or standardize the quality of the pathologies. Therefore, while the molecular, just can give us some update on the more basic pathology tasks, whether the AI the application of AI in this area.
Yeah, there are quite a number of publications to find the tumor, and but the only one that is approved is at the moment for prostate cancer from AI. So there isn't anything approved for the GI tract. There are a number of publications, identifying the tumor, also identifying the tumor grade, identifying the depth of the invasion. So people are working on it. But similar, in that respect, it's more or less similar to molecular studies. People don't see it through. So like we've done all this effort from one year to the next, increasing the numbers for the microsatellite instability studies to really push it through. Those studies, how to find the tumor, how to diagnose the grade, etc., people publish it, but then nobody picks this algorithm up and does it again, like you would do in a proper study. And so that's the danger now, that everyone is just pushing out one paper, and then they just move on. And I think that that's then similar, like with all the molecular markers. These markers will not become proved and will not come into the routine. You need to have a similar strict protocol for validation for these methods like for molecular studies. It works, yes, but it hasn't been done on these large numbers, and it hasn't been validated. But clearly for also, let's say, PDL1 scoring and those things which are very difficult, that all works with AI, but it has not been consequently tested in large populations.
Thank you.
Beautiful talk and very thought-provoking. I wanted to follow up on that comment from my colleague here about the heterogeneity. If you have a tumor that's 99% in situ disease, but there's just a small little piece that's actually invasive, and you're choosing which areas are going to be sampled for the molecular testing, it's a little different use of resources and choosing how to do that than when you're scanning it visually over the course of the whole field. What is the conversation around how to standardize that aspect of it, so that you're not giving a different molecular diagnosis than perhaps what the pathologist can see when they select out just a few specific areas that may actually be the highest level of dysplasia or cancer that could otherwise be missed?
I mean, there is no standardization for that, but I think it's necessary to really do that.
What's the conversation around how to make sure that happens? Is it on a grid? Are there a certain number of fields? I'm sure this is an area that has to be really vetted out for this to move forward.
Yeah, no, I think so. I would agree, but no, that hasn't been. I mean, there's a lot of work basic work that needs to be done. It starts with the standards of the imaging of the slides, which magnification you're going to use, which scanner are you using, what quality control are you doing. All of these has not been sorted. At the moment, there is an enormous pressure that people are just trying to get their papers out. And those basic aspects, they are just forgotten. But they are clearly necessary, I think.
Well, it's very exciting. Thank you.
Thank you. Thank you, Professor Heike. Yeah, in the interest of time, we'll move on. Thanks, Professor Heike, for your very interesting talk.